Hello, everyone. Welcome to the first MIT Colloquium this semester. Today is a great pleasure and honor to introduce my dear colleague and friend, Cullen Buie. Cullen is an associate professor in MIT Department of Mechanical Engineering and director of the Laboratory for Energy and Microsystems Innovation. Cullen received his PhD from Stanford, followed by a postdoc at Berkeley, and then he joined MIT. He has been honored with numerous awards, including NSF Career DuPont Young Investigator, DARPA Young Faculty, NSF Presidential Early Career Award, and in 2021, he has been elected to the Fellow of the American Institute for Medical and Biological Engineering. Particularly noticeable, Cullen co-founded Kytopen, a startup company. The company has been backed by MIT Engine and has raised over $40 million in funding to today. And I've been chatting with Cullen. So it seems the company now is partner with multiple other companies pushing multiple products through FDA approval. With that, I give you, Cullen.
All right. All right, so I'm very, very happy to be here. Thank you all for coming. I'm going to be talking about really two different journeys. So I'm going to talk about some work happening in my lab that's been dealing with genetic engineering of bacteria. But then as Juanha, as he mentioned, I'm also going to talk about some work happening at a startup that spun out of my lab about 6 years ago in 2017. I actually spent a considerable amount of time there. I took 2 years of leave from MIT while it was starting, and I'm still relatively involved. So I think, as you'll see, there's some pretty good parallels between some of the work happening in my lab and some of the work happening at this company. But let me first start with some motivation for particularly some of the things happening in the lab. I got into these spaces really thinking about the bacterial and the microbial world, and you might say some of the limitations we have in tools for exploring it. So to put this in, to use an analogy, let's say that this map of the globe, let's say that this represents all the bacterial species on the planet. So we don't know the exact number, but I'm curious, who could guess what are some of the numbers out there that people guess how many different species of bacteria are on the planet? It's more than 10. Any guesses? Don't be shy, I'll call on somebody. Yeah.
Maybe 10-6.
Yeah, so that's a pretty good guess. So it's guessed around 10 to the 7 or more. So let's say that this globe represents all of those 10 to the 7 species. Now, how many of those species of bacteria have actually been isolated, characterized by humans? We're talking about 10 to the 4th. Okay, so that's like saying roughly maybe the area of Alaska or less. So imagine if on our planet, all we had characterized was just Alaska. But I'm also interested in gene delivery. So of these organisms, how many can we genetically manipulate and use for genetic engineering, or just simple a kind of fundamental understanding of genes? It's about 1/100th of that. So on the order of hundreds to maybe thousands, right? So that's like an area the size of Connecticut, right? So imagine if on our whole planet, all we had access to was Connecticut. So there's a lot of world out there. So this problem, all these genes that we don't have access to, all these organisms we have limited ability to engineer, this is kind of this known as this area of genetic dark matter, microbial dark matter. So there's just so many genes out there and organisms that we don't have the ability to touch. Now, why are we interested in this? Because for those that we do have tools for those that we can engineer, we can do a lot of things with them. You just think of one, E. coli, it makes on the order of $20 billion a year in various, industrial and pharmaceutical chemicals. And this is actually an old number. This is a few years old. So the thing I always think about is how many other organisms like this are out there on the planet that we either can't culture or can't genetically manipulate. Now, related to that topic of genetic manipulation, when we think about genetic manipulation, we're not only interested in engineering cells to get them to produce something, we're also very interested in understanding how they work. So the explosion in genetics has allowed us to develop, us being the research community, various tools that allow you to probe how genes, how different genes lead to functions in a cell. Kind of one of the, you might say one of the classic things to do is you take some organism, you build a library of mutants, and then you see how those mutants respond to different stresses, either in their environment or in growth. And so you can determine, well, what genes are essential and what genes might lead to fitness enhancements. Now, I've been thinking about this for a while. So, one of the things that happened, we've been able to read and write DNA and reduce the cost of reading and writing DNA at an impressive rate. So this is a graph from the NIH, from the Human Genome Project. And what you see in the white, that's Moore's law. So if the speed at which we were able to reduce the cost of reading and writing DNA were following Moore's law, it would be that white curve. And what you can see is that it's actually gone much faster. So one of the things that's happened is that the biotech community has moved extremely quickly. And one of the things, the results of that, we have actually been slow in developing tools to use the advances that we're developing in molecular biology. So we have been able to read and write DNA at impressive speed, but there's been a bottleneck, and that bottleneck has been delivery. So we can read and write DNA, and we can conceive of a lot of different things, but we can't get it into all the organisms that we would like, or into all the cells that we would like. And so we have to catch up. The engineers and the technologists have to catch up to these advances. This is perhaps maybe, this is very evident in the area of CRISPR. So many of you have heard of CRISPR. What is CRISPR? CRISPR is just one of a few, but easily these days the most popular gene editing technology. So what is CRISPR? CRISPR is a package. CRISPR is a package that you deliver into cells that allows you to edit them. And there are many different packages or many different flavors of CRISPR. I'm just showing a couple of them here. CRISPR-Cas9, which is maybe the first one that was discovered and most popular. You deliver RNA with a protein that allows you to find a specific sequence of double-stranded DNA, break that double-stranded DNA, and then perhaps insert something into the cell or allow the cells to repair its own DNA, and this can lead to some kind of edit. But there are other things you can do with CRISPR. These days you can do things like base editing, and so you can edit a specific part of the genetic sequence. You can also silence a gene, or you can upregulate a gene, but this is just a package, right? A package that isn't delivered, it has to get into the cell before it can do anything. So the analogy that I like to think about here, show of hands, how many of you have Amazon Prime? So almost everyone has Amazon Prime. If you're like me, have you ever put a bunch of things in your cart and forgot to hit checkout? And then forgotten that you didn't hit checkout? And then you're like searching at your door like 2 days later, like, where is my package? And then you go to Amazon, it's like, oh, I never had it delivered, right? So having CRISPR, but not having the ability to deliver it into cells is like having things in your Amazon cart that you never checked out, right? It's useless. It's useless until it gets in the cell where it can actually do this action. And you don't have to take my word for it. So this is a quote from Jennifer Doudna. Delivery remains perhaps the biggest bottleneck in somatic cell genome engineering, a reality that has motivated increasing efforts across disciplines. So until we're able to get all of this information, all of these constructs into cells, we won't fully realize the power of gene editing tools like CRISPR and other things. So my lab does a few things, but one of the things, and I'm only going to talk about this story today, is we seek to enable genetic manipulation of all types of cells and all types of organisms. We're going to talk about bacteria and human cells today, but the ultimate mission is to enable these advances in genetic engineering across biology. So just two parts of this talk. The first talk, I'm going to talk about some things happening in my lab where we've been advancing tools for delivering genetic material into bacteria, some tools for large volume delivery, and for high throughput parallel delivery. And then I'll talk about some companion things that have happened in parallel at the startup Kytopen that spun out of my lab about 6 years ago. So a lot of the work happening in my lab has been done by a research scientist in my lab, former postdoc, Po. Is Po here? Po's here somewhere. Oh, there he goes. Okay. So he made this great video. So one of the things about bacterial, one of the ways that genetic materials delivered into bacteria today is something known as electroporation. So you mix a batch of cells and the payload that you want to deliver together, you put them in a cuvette, and then you have to zap them in order to deliver the material into the cell. When you apply this high electric field, pores open in the cell temporarily if you apply the right mix of field and the buffer conditions, and the foreign material can get in. The problem, though, is that this process is very manual. And so I asked Poe, well, how fast could you do it? If you were just using the current technologies, how long does it take? So this is at 5x speed. He made this video. He added the music. I love the music, actually, because it is kind of ridiculous. Like, you could maybe do on the order of one of these every 2 or 3 minutes, if that's all you did. So all you did for an hour, maybe you could do 20 of them, maybe. So you have to wash the cells, you're aspirating back and forth, you shake them up, you put them in the system, you zap it, you gotta take it out, you gotta pipette again. I think we could do better than that. Okay, so cuvette-based electroporation. It's pretty simple. Some of the pros of electroporation, you can deliver a wide range of materials, both large and small molecules, which is great. It's not cell type dependent. So there are many ways of delivering things into cells. You can use viruses. You can use, in some cases, you can use nanoparticles. One nice thing about electroporation, it's cell independent. So you make one system and you simply change the electrical properties, and that changes the type of cell that you can deliver to. So you don't need multiple systems for multiple types of cells. That's a benefit. And it also can be highly efficient. But there are some drawbacks. You're limited in the volume because it's a batch-based process. The batch, because it's manual, takes a decent amount of time, a couple minutes. And because of this, it's very operator dependent. It's very manual, very slow. And so two people doing what they believe is the same process can get very different results. Now, there are some applications. Early on, when motivating, I talked about how one of the great things about the advances we made in genetics is that we can use molecular biology techniques in order to create mutants where different genes are knocked down or knocked out. And from those mutants, we can understand how those genes function. Now, cells, if we think of bacteria, can have on the order of 2,000 genes. And so that's a lot of different genetic transformations that you might require. Or you might need a lot, or you might need to deliver this to a lot of material. If you're limited in volume and limited to something manual, it can be hard to make these mutant libraries. Also, another thing, many organisms are hard to transform in the sense that E. coli and many strains of E. coli are relatively easy. When people think about this problem, they often think, oh yeah, bacteria are easy, but when they think that, they're usually thinking E. coli. They're thinking about one specific organism. When you broaden and think about that whole planet, there are many organisms on the planet that are actually difficult to transform, to genetically transform. And so if you have low efficiency, you might need to use a large volume in order to get a sufficient number of transformants. And the batch-based electroporation system really is limited in allowing you to do that. So we thought, well, maybe we should do something that's flow through. It turns out the cells don't have to stop. Why don't we let them continuously flow? And there are some flow-through systems that are in the literature. One of the challenges, they're not very scalable. They also, all of these examples, most of the work has been done in human cells or eukaryotic cells, not in bacteria. It turns out it's more difficult to deliver things into bacteria. So a quick scaling here. When you're doing electric field-based delivery, the size of the electric field required to open pores generally scales with the size of the cell. So a smaller cell actually requires higher field. So if you reduce the cell size by around a factor of 10, you generally need about a factor of 10 higher electric field. And all of these systems were designed for mammalian cells. And so they're not applicable to bacteria. So they don't scale very well, and they don't work well with bacteria. So we needed something new. And I guess one thing I will note here, we wanted to design something that would not just scale, not just work well with bacteria. We wanted something that would be easily deployable to other labs, and we wanted something that you could imagine could work in high volume very simply. We didn't want to have a system that the only people that could use it was us. We wanted it to be very simple. So keep that in mind as you see what we've devised. So we wanted something minimal fabrication, scalable, low cost. It has to be able to be used with bacteria and requiring minimal sample prep. And so we devised this platform, which isn't even microfluidic. I mean, we're saying it's a microchannel here. It's not even really a microchannel. This is a piece of tube. This is about as simple as you could possibly imagine a system for doing electroporation. But we found out, why does it have to be complicated? It doesn't have to be complicated. All you need is an electric field and a flow path. It turns out that the residence time within this system is on the order of milliseconds. That's all that's required for the cells to be exposed in order for pores to open. So right now, if you recall that last video or the video from Poe, it takes you roughly 90 seconds to get the cells into the cuvette, and then you zap them and you get them out of the cuvette. They only experience the electric field for like 5 milliseconds. So there's no reason for them to sit in the cuvette for 2 minutes. So I'll give another analogy here. I love analogies. 20, 30 years ago, you drive through a toll booth, you have to stop, right? You pull your change out of your pocket, you throw it into the thing, hopefully you have exact change, and you don't need to like get change. And then the thing opens up and you drive through. How do tolls work now? They don't work like that. You have a little transponder on your car, you drive up, automatically they just take the money out of your wallet and you don't even stop, right? That's how this is working compared to previous gene delivery. There's no need to stop. Why do the cells need to stop? So the throughput is much higher, just like the traffic throughput is much higher with the toll booth where you can continuously drive through. So, as mentioned, very simple system, very cheap system, very easy to fabricate. No mechanical machining required in order to devise this platform. It's also compatible with syringe pumps. We wanted it to be something that you could easily deploy in almost any lab. And a syringe pump is a very, very low cost, relatively low cost, and widely available piece of equipment. So let's look at some of the data. One thing you might wonder, so we're actually applying relatively high flow rates. So the flow rates in our channels or the Reynolds numbers on the order of 1,000. So they're moving relatively quickly. You'll see some of the flow rates we have anywhere from, half a meter per second to a couple meters per second. So these cells are moving very quickly. So one question is, does that in and of itself open pores in the bacteria? And so what you can see is that this is a plate. What we're showing, the plate is selective. So any cells that don't have a resistance marker for the antibiotic don't fluoresce. Those that do have the resistance marker will fluoresce. The only way they get the resistance marker if they were successfully genetically transformed. And so in the top is where we're just flowing and there's no electric field. So if we flow and there's no electric field, we don't get delivery. And so you don't see cells growing, you don't see the fluorescence. When we apply the electric field, you can see that with the flow, we do get genetic transformation. One thing to note here we're comparing what happens in the flow system with what happens in cuvettes, right? So cuvettes, here, these are just the electric fields used for cuvettes. One of the things to note, we're using this electric field. So we're using a lower electric field, but we're getting generally higher efficiencies than what you see in cuvettes. And one of the things we're working out, and we believe, is that there's actually a coupling between the flow and the field. So the flow by itself isn't enough to deliver, but the flow plus the fields actually reduces the amount of electric field that we require. And so you're going to see this throughout. The electric field that's required in cuvettes is consistently higher than what we need in the flow plus the cuvette. Okay, I'll say that again because I see some confused faces. So what's happening? We're delivering energy into the cell. That cell can come in many forms. It could be mechanical energy through the stress of the flow. It could be electrical through the electric field. We are delivering mechanical plus electrical, and so we need lower electric fields than what you need with just the cuvette by itself. One thing to note, and I just mentioned this is a log scale, so you can see that the efficiencies that we get for the same electric field, this 8.33 kilovolts per centimeter, it's anywhere from 1 to 2 orders of magnitude higher when we add the flow to the process. I won't show all the data, but we've demonstrated this on multiple strains of E. coli. E. coli NEB10 beta is kind of a workhorse strain for synthetic biology. It's very highly transformable, so you tend to see very high transformation efficiencies. And once again, similar to before, you can see at the lowest field strength we use for the cuvettes. The cuvette data is to the right of the dotted line. At that field, we are getting significantly higher transformation with the flow M-tube device, our flow-based system. We also tested this on E. coli MG1655. This is just an environmental strain that we wanted to test, not a lab strain. Interestingly, at this low field, you've got almost no transformation in the cuvette, where we're still getting substantial transformation in the flow-based system. Now that's interesting, but is it useful? It can be useful. So recall earlier, it's part of the motivation, we said, well, one of the reasons people want to deliver things into cells is they want to tell, well, how do the genes in those cells work? One of the ways that's done is you can deliver what's known as a transposon library into the cells. So these transposons will integrate into random parts of the genome. Okay, and what you would like to do, you'd like to get these random insertions into the entire genome so that you know that you have knocked down every single gene within the cell. And so then what you can do, once you've done that, you can put it through some kind of fitness assay and determine, well, which organisms had trouble growing after this stress, and that helps you determine which genes might have been related to allowing the organism to survive that stress. And so we didn't do that second experiment. What we did do was just deliver the transposons into the cells in order to determine if we could make a library and perhaps make it more efficiently than what you could do in cuvettes. So the first thing we tried, we delivered a plasmid just to optimize our transformation conditions. And what you see here, the plots for each bar, the left plot is the M-tube R system. The right plot is with cuvettes. And what you can see, we can get comparable or much better, in some cases, transformation efficiency compared to the cuvettes with the plasmid. Then we use these conditions. The conditions here, we were running at around 0.6 meters per second. So once again, the velocity, the linear velocity is reasonably high for something that people might consider microfluidic in these channels, and 8.33 kilovolts per centimeter. And we delivered the transposon into the cell. And once again, we're still getting better, higher efficiency than cuvettes, but we're doing it much faster. In the cuvettes, you can only do it 100 microliters at a time. We could do 10 milliliters in, less than a minute. Okay? And then what you see on the right is some preliminary results of this, and this has been published, where we're delivering into a particular organism, B. longum. So B. longum, it's an organism that's in the gut. It's being explored for probiotics in the human microbiome, and people would like to understand how this organism will respond to various stresses, what genes are most important. And so we were able to create a library where we saw 2,000 unique insertions across the genome of this cell from our delivery. And so this is the type of study that you could imagine doing in an organism to understand how those different genes, once you've done the insertion, you essentially inactivate that gene. And so you can understand how the different genes might correspond to fitness and other aspects of this organism. So this was for large volume. Okay, we're doing large volume transformation of bacteria. Now there are some cases where you're not necessarily interested in large volume, and maybe the thing that you want a lot of, maybe you want a lot of different experiments. So if we had to run 1,000 different experiments, this wouldn't be very convenient. If you wanted to run a large volume, this would be. So what do you do if you need a lot of parallel experiments? We actually are going to continue to use this concept of continuous flow coupled with electric fields, but in this case, we're going to do it in a parallel manner. So we created this concept for automated high throughput parallel genetic transformation, in which case we take what was happening in that previous system, and now we have this layer by layer system in which you have a channel, 2 PCB-based electrodes, a seal layer and a seal layer. but you have 96 of them in parallel. And so you can operate this system very easily on an automated liquid handling apparatus. And so now you can take the user completely out of it. They just have to program the system, and you can run a 96-well plate in an automated manner. And so you can do highly parallel transformations using the similar physics as we showed in the large volume system. This is one of the real images of how the system looks. Something to note, we're always pushing towards trying to make these systems scalable and ultimately deployable down the line. So we're thinking about how it can be manufactured. Many of the parts of this system can be injection molded. The electrodes are made out of PCB. We anticipate with our current design, each one of these, just the fabrication of it would be around $20 each. So if you want to compare that to cuvettes, a cuvette where you do one experiment is around $3 or $4 each. And so we're doing 96 for around $20. That's like the vision that we're pushing for here. And so you could imagine we want to have these things available so that people could do hundreds of experiments in a day, and the cost is not a big barrier. So we worked with an engineering firm also named Fix to make this happen. And so we designed the initial proof of concept, and then they helped us make a system that we felt would be kind of scalable and repeatable. You can take this is another image or another version of that electroporation plate, that 96-well electroporation plate. We can interface it with electrodes, and then we mount it in our lab on an automated liquid handler. One thing to note, this system is universal, and so it's not designed for one particular automated liquid handler. The way the seal is designed is you can use pipette tips from anybody's automated liquid handler. And so we also wanted it to be, it doesn't matter if you have a TCAN or a BD system or an Opentrons, it should be compatible with all of them. So here's the system in action. So what you can see, it'll pick up the pipette tips. You can see the syringes in the back. Now it's picking up sample. It's going to go down. You probably missed it, but there were pulses that were delivered. Once it comes back, it's going to pick up sample. It's going to go down into this system, which is where the electroporation happens, and you're going to see pulses applied. And so in this case, it's doing 8 at a time. This is at about 2x speed. The system can do a 96-well plate in about 5 to 10 minutes. And so you compare 96-well plate in 5 to 10 minutes compared to the manual pipetting. There you're doing like 1 in 2 or 3 minutes or more, and your poor hand, right? So we've used this system for a few things. This is all unpublished, but in process. One of the things you could imagine doing with a system like this is you want to screen for field conditions. Let's say you have a new organism, you have some new hard to genetically engineer organism, and you have no idea where to start. You can do 96 different transformation conditions, once again, in about 10 minutes. Right? So we've used this. In this case, we're screening for field conditions, and what you can see as we move from relatively low fields, 1 2/3 kilovolts per centimeter, up to 18.3, you can see we slowly start increasing in efficiency, and then at some point, you start to see it fall off because you're killing all the cells. Right? So there's a sweet spot, right? You need to apply enough electrical energy so that you open pores, but not so much that the pores are so large that the cells die. But you can screen those conditions very easily using this platform. You can also screen flow rates. So here we're showing different flow rates. This is in microliters per second. The main point being you can screen fields, you can screen flow rates, all of this very easily using this platform, fully automated, no need for the user to do all that pipetting. So you could imagine, hypothetically, you could use this system to screen for the flow conditions, and then if you want to scale up to something larger, you could use the previous system if you wanted to genetically transform a large volume of cells. And the system is pretty repeatable. For something we are making it even more repeatable, but we actually feel pretty good about where it is. Here you're just seeing for different field conditions along different columns how the transformation efficiency, so the number of transformed cells we get, the efficiency is defined as the number of transformed cells you get per microgram of the payload that you're delivering. And you can see that it's relatively flat across the columns. Because that's, of course, if you were doing something perhaps more complicated, let's say you're delivering the same thing, or let's say you're delivering different things in each well. Let's say you're delivering a different CRISPR construct. You want to know that each well is getting the same, getting a predictable performance. Otherwise, you might attribute something to the biology that's not really the biology, it's your system. Right? So right now, I'd say our first generation, we would get maybe 1 or 2 errors per plate, but our vision is to have more like 1 or 2 errors per 100 plates. So that's where we're trying to go. Okay, so I'm going to summarize here before moving to the next session, next portion. So we have these two kind of parallel complementary platforms. One for large volume delivery of genetic material into cells, one for highly parallel automated delivery of genetic material into cells. Both of them use a combination of flow and electric fields to open pores in the cells, and to do so in ways that are superior to the state of the art, which is cuvettes. If you think about the system on the left, this is for large volume, much larger volumes than what you would conceive of doing with cuvettes. So our flow rates are order tens of milliliters per minute. So compare that tens of milliliters per minute to doing roughly 100 microliters every 2 or 3 minutes with cuvettes. So it's orders of magnitude faster when you need to do large volume. Conversely, the high throughput system, you can do a 96 well plate in like 5 to 10 minutes compared to doing 1 or 2 transfections in 5 to 10 minutes. So this is also several orders of an order of magnitude or more faster than what you could imagine a person doing in an automated manner. So what's interesting, I'm going to transition here. All of this work was in my lab in bacteria. About 6 years ago, we started a company. Initially, the company was going to work on bacteria, but quickly realized that there were some perhaps more interesting problems in human cells, or perhaps more lucrative, maybe not interesting. We'll say more lucrative problems in human cells, and took a parallel path, but in delivering genetic material into human cells, and I'll talk about that now. And so that's Kytopen. So I'll say for students and postdocs who are interested in the audience, we were one of the first 6 companies funded by the engine. So the engine, everybody knows what it is now, but when we were there, it was like a brand new thing. So we were one of the first companies funded by the engine. I've been pretty involved. So for those interested in either the engine or entrepreneurial activities in general, I'd be happy to talk to you more after the seminar. So here we're not thinking about bacteria, we're thinking about the industry of cell and gene therapy. Show of hands, who's heard of cell and gene therapy? Okay, so maybe 60, 70%. So I'll give a really quick explanation, just make sure we're all on the same page. So cell and gene therapy, in particular, we're thinking about cell therapies where the motivation is to take human cells, either from a patient or from a healthy donor, and give those cells to a patient in order to cure or treat their disease. In many cases, those cells would be engineered. So you're going to take a cell, you're going to engineer that cell in order to affect a positive clinical outcome on a patient. I'll give a quick example. If you think about cancer, cancer affects many people. It turns out that our bodies all have precancerous cells. Like there are precancerous cells all the time. Normally what happens, our immune system finds those cells and gets rid of them. Like the normal process of things is our body finds those precancerous cells, and whether it's NK cells or T cells, recognizes them and gets rid of them. You could say in some ways that what happens in cancer is that those cells have figured out a way to trick our body into letting them live. And so then they start to proliferate and can cause issue. There are therapies that are being developed now where you kind of want to retrain your immune system to find those cells. I might even describe it as a software update for your immune system. So the way your immune system normally works, it finds those cells, gets rid of them. In cancer, that gets hijacked. In these cell and gene therapies, what they want to do is retrain your cells through genetic engineering to then find those disease-causing cancer cells. There's a lot of activity in this space, roughly 2,200 clinical trials, 60% of them focus on cancer. And as said earlier, though, delivery, as Jennifer Doudna mentioned, as I talked about earlier, it's a major bottleneck, not just for bacteria, but also in this field as well. There are various payloads that are looked at being delivered: mRNA, DNA, CRISPR for knockout or knock-in, plasmids. And basically, as you are developing a therapy, moving from the bench all the way to the clinic, delivery is one of the few things that goes along with you. I'll tell a quick story. CAR T therapy, one of the very first approved CAR T therapy was by Novartis, which they licensed from University of Pennsylvania, a drug called Kymriah. Who's heard of CAR T or Kymriah? So a few hands there. So this is an example of what I was talking about earlier. So you engineer what's known as a chimeric antigen receptor into T cells. And those T cells now have enhanced activity against blood cancers. Now, what I've heard from people in the industry is what Novartis did is they took the process from UPenn and they just immediately scaled it up. Because in cell therapies, as the name suggests, the cell is the therapy. So everything that happens to the cell is going to affect its outcome. And so there was a hesitancy to change anything about the process as you moved from the bench all the way to the clinic. And one of the challenges that they had is that some of the initial processes used at the bench, when you were only doing a couple patients, didn't scale very well. And so there were major challenges in the field as you scaled up from the bench to the clinic. And so when you decide how you're going to do your delivery, this is one of the processes that is going to scale with you. So you need a scalable delivery system. Just because something works at the clinic or at the bench scale, it has to be able to scale up. Otherwise, you're going to have problems as you move to the clinic. And this has already been shown in the field. One of the ways that this has kind of reared its ugly head is that there's been just major problems in manufacturing. So one of maybe the biggest bottlenecks is figuring out how to manufacture these therapies, which, as you can imagine, for many of the researchers in this area is pretty frustrating. So you have a therapy that works at small scale, but the challenge is in how do you scale it up and deliver it to people in a reasonable amount of time and at a reasonable cost. So why does it take so long? Why is it so expensive? A lot of this is just related to how drugs are developed in general, but as you can imagine, with these complicated cell therapies, it's ratcheted up even another level. At the early stage, it takes hundreds or thousands of experiments. You need hundreds of experiments in development. This can take anywhere from 2 years in preclinical, 3 years in the early clinic. Manufacturing can take a long time to scale up. It's order of $1 to $2 billion for developing one of these therapies. Now, we think that one of the ways that this could be cut down is by making the discovery process faster and making the various interfaces faster. So we want to be able to discover the drugs faster, and we want to be able to scale up faster. So this requires a technology that allows you to do many experiments in parallel, so you can do high throughput discovery, but that same technology has to be able to scale up quickly. One thing I'd like to note, I alluded to this earlier, because the cell is a therapy, early decisions you make in how to process the cell actually propagate as you scale up. So you need to make decisions about scale up actually long before you're ever going to get into the clinic. And so you need a technology that will scale with your therapy. What we believe is that this technology, which once again has a continuous flow method for gene delivery, will cut down the number of experiments, mainly because you can do a smaller number of experiments but in high throughput. Right? So you're going to do a smaller number of experiments but in automated high throughput. So you're still going to generate just as much, if not more, data, but in a shorter amount of time. But because the technology is continuous flow, the throughput is not an issue. As you need to scale up, all you do is change the platform. You don't have to change anything about your test conditions. So imagine a system in which, while you're doing the discovery, you've actually already designed your scale-up properties. It's like you're doing two things at once because it's the same technology that's used at the small scale and at the larger scale. So how does this work? The main thing is that the technology that's used for delivery, this is the automated high throughput discovery scale system. This is the scaled up system for manufacturing. So I'm showing a schematic here, but these are actually real products. I'll show them to you later. Both of them leverage the same physics for delivery. And the physics are the cells are moving, the cells never stop. They're mixed with the payload. They experience an electric field while they're in this small region. And when they experience that electric field, pores open, and you can deliver material into the cells. So this transfection apparatus, it's exactly the same in the two systems. So we manufacture a bunch of these, and when you manufacture them, you don't know if they're going to be used on discovery or for the clinic, because it is literally the same. It's not like the process is like the same. It's literally the same. You see the video on the right. You can see the technology is a little different than what we were using in my lab. Here, the transfection occurs in this apparatus, so as opposed to having the genetic transformation occurring in the 96-well plate, it's actually engineered into a pipette tip. Right? So you have these specially engineered pipette tips with electrodes. When it moves into this box, in this, that 5 or 6 seconds that it's sitting in there, it's delivering material into cells as the cells are being aspirated into the 96-well plate. So this has been used to enable CRISPR and actually delivery many other things into human cells. So I'm going to show this plot. First off, this is a normalized plot, so these are not absolute values. It's normalized based on a standard condition from our system, but then also, if you see this triangle, sorry, I'm not tall enough. You see that triangle? That is a commercial in the clinic electroporation system. So we're comparing our technology to commercial electroporation technology, which once again is batch-based. Okay, so we're comparing it to a batch-based system. And what you can see, we can run, a couple hundred experiments in under one day to optimize the process. Okay, so we can optimize, as talked about earlier, in less than a day. And we can see that we have several conditions where we're able to get higher viable cells. Okay, so just normalize the 1. So when you see a 2, we're getting twice as many viable cells. So there are cases we're getting twice as many viable cells. You look at the axis on the left, that's the normalized efficiency. So we're getting twice as many viable cells, and in some cases, 15 to 20% more editing efficiency. All in a system that's continuous flow and high throughput and amenable to the parallelization. So this has been done in several cell types. T cells. T cells are very interesting for cancer. We've also done this in CD34 positive hematopoietic stem cells, which are useful for blood disorders, but also for cancer. B cells, NK cells, many of the cell types that are being explored for various engineered cell therapies. I'll explain all these plots. Here, we're delivering mRNA. So mRNA is useful for some therapeutics. In some cases, you don't want to have something that is permanently edited. You'd like it to be transient, and so you deliver mRNA. And what you see, the open bars are no transfection. Okay, so no transfection. The closed boxes are with the transfection. So you see there's negligible hit in the viability. So comparing nothing to the system, there's negligible hit. But you are getting delivery. And so looking at the efficiency after the mRNA delivery, you're getting order of 90+% efficiency from that delivery. But we've also looked at RNP delivery. So this is ribonucleoproteins. So this is a CRISPR construct. You take the CRISPR RNA and the Cas protein, and you fuse them so that it's easier for delivery. And you're delivering this for gene editing, often for knockouts. So we've done these knockouts in T cells, NK cells, CD34s, and you can see still high viability, 80+%, and efficiency, knockout efficiency, also on the order of 80%. One of the interesting things we saw is this is comparing the system in T cells to a commercial electroporation platform, and this is a population doubling level at 48 hours. One of the things we've suspected is that because we're having flow plus electric fields, we've already shown that we can reduce the amount of electrical energy that we need to send to the cells because of the flow. The cells seem to respond and recover better. So the cells are doubling faster. So you're getting higher efficiency, you're getting higher viability, and they seem to be recovering faster in some cases, which, as you can imagine, could be useful for a therapy. Okay, so I won't spend too much time on this slide, but very excited about where this technology is going. It's kind of cool to see what's happening in my lab and what's happening at the company. I've been pretty involved in the company, as I mentioned, spending like a day a week and a couple years off from MIT. So it's kind of cool to see both of these things moving forward. One for the case of cell therapies and enabling these engineered cell therapies, and another in the case of bacteria. But I'll end with this note. Maybe some motivation for me. So this is my son. Sorry. So he has sickle cell disease. And I guess what I wanted to say is when we started Kytopen, I mentioned we were working on bacteria. And later on, we actually transitioned to looking at human cells, which is like, I knew nothing about it. So he's 10 years old. So for the most part, he's actually doing okay. He's 10 years old. He's got sickle cell disease. He's mostly doing okay, but he does have the disease. And I find what's kind of cool and just really something enabled by being at MIT. It's like we start this company, and because we're in the area, people start telling us like, hey, you got this technology for gene delivery into cells. You guys should be thinking about cell therapy, right? And like my lab, as you saw, we were doing bacteria. Like we weren't thinking about this at all. But because the technology is so simple and so scalable and works so well, we were able to like really easily change over from looking at bacteria to looking at these human cells. And then so I find out later, like, wow, this technology that we're working on, which can engineer CD34s, could help kids like him. And yeah, I realize I'm tearing up. It's mostly like excitement. So to be clear, it's not so much the sadness. It's like just like really cool to like do work. And really like feel that kind of impact. So I need to stop. I want to thank a lot of folks. So our research lab, this is a research lab and kids just at a Red Sox game. This is Kytopen. This is Paulo Garcia. So Paulo was a postdoc in my lab, then a research scientist. He was very industrious, very industrious. He was in the Translational Fellows program, which was this program that MIT had for a minute to help postdocs start companies. He got us connected to the engine. He was CEO at Kytopen for the first 6 years. really helped us build this, get it to where it is. Mentioning some lab alums, definitely want to thank Poe. So Poe did, like, he was involved in all the bacteria work that you guys saw. Several collaborators and funding sources. So sorry for all the emotion at the end, but I couldn't help it. Thank you so much.
Great. Questions? All right.
So for transfecting human cells, is there a payload size maximum that you can, or can you go like way up? Because I think that's also one of the problems for delivering genetic payloads to eukaryotic cells.
Yeah, that's a great point. So there is a limit. I don't know, we don't know the tip top, like what the actual physical limit is. I mean, we have seen that as the payload starts to increase, you do see a decline in the efficiency, and you start to need a higher electric field, which then leads to a decline in the viability. We have seen that we are generally able to deliver larger payloads than what you might be able to deliver with AAV. So if you know anything about, AAV, adeno-associated viruses, they're around 4.8 kilobytes. Kilobases, I'm sorry. And so we can do significantly more than that, but we haven't found like the real like upper limit. But for sure, larger payloads is an issue. So it's not as much as an issue as it is with viruses, but there is a limit on there is an upper limit. So you can't just like deliver a whole gene or something like that in there. Does that answer your question? Kinda?
Can you, very nice talk, Colin, can you give us a perspective on where, for the bacteria side, phage fit in? Because phage are useful for some things, but not everything. And I guess for like treating patients who have some pathogen, you might do. So can you just give us this really short Cliff Notes version on where transfection of bacteria by phage fits in the whole landscape with what you're doing, because you can transfect them.
Yeah, no, that's a great point. So phage are great when you have a phage that you can engineer for the organism. So
you need to know the phage.
You need to know the phage, and phage is they're very specific.
Yeah.
So they're species and even and strain specific. So when you have a phage, you would do that. If you have one, you would do that. But in many cases, you don't. And so the advantage of, I would say, any physical method. So I'm going to say, we're doing a flow-based electroporation. But really the advantage of any physical method is its ability to perhaps have a broader, a more diverse array of organisms that it can act on.
So you mentioned that your device works in 96-well plates. Is there any push towards, or is there a need for 384 versions, or like 1536 plates?
Yeah, that's a great question. So ironically, what we found, one of the things we found is that many customers can't handle 96. So the things seem to be almost bimodal. Like there are groups who are still doing everything manually. And then there are other super high throughput groups that are trying to do 384 or 1536. And so there is a need for larger, but it's a very, very, very small market that are actually, because if you think about it, what is the challenge? You're going to do this high throughput delivery, but then everything upstream and everything downstream has to also be high throughput. So you really need to be a team that has a workflow that's already suited to doing high throughput science. And in the cell therapy space, that's actually not everybody. So we've come up with some creative ways of helping customers to get access to the high throughput nature of it. But many of them, they look at the 96 and are like, wow, that's awesome. I don't know how I'd use it because I'm doing everything manual today, right? And so some of it is dependent upon, as high throughput sciences, kind of continue to penetrate this field, that will help. So I would say we've heard that, but it's from 1% of potential users.
Cool. And another thing, so I guess it's a completely different question. But like the thing that you mentioned about the cells doubling faster upon electroporation, is that faster than the control? Like, is the rate just higher since it's electroporated? Like, the cells grow faster, or is it because of you've also transfected them? Like, what attributes the increasing growth?
Yeah, that's a good question. So it's faster than another electroporation. So it's faster than a commercial. I won't mention the name of the vendor, but there are only a few of them. It's faster than a commercial electroporation vendor. So both of them are getting delivered. Both of them are getting electric fields. What's a bit different in our case is they're not just getting electric fields, they're also getting the flow rates, which can reduce the electric field. We have some preliminary evidence from transcription analysis suggesting that the cells are less stressed after this process compared to just a pure electric field delivery. And so we believe it's just because the cells they're happier. But that's still work under development, but that's the hypothesis.
Yeah, so for the first part, did I? Oh, go ahead. No, go ahead.
Go ahead, start.
Okay, for the first part, for the genetic dark matter, and to kind of go out into a larger cross-section of the species, what are you, like, I'm trying to understand what the barrier is. Do they not get porated, or do they get porated but not have low viability? And then, are you thinking that, like, flow plus field, there might have to be other physical stressors too to kind of get a wider, just tell me a little bit what you're thinking about, like, to go, like, onto huge numbers of species.
Yeah, so that's a great point. So I'm thinking at least two or three things. So one, we have a collaborator at the Fred Hutch, Chris Johnson, where we've been working together for a few years and thinking about this problem. He's coming at it from the molecular biology perspective, where many organisms have, restriction modification systems where any foreign DNA comes in and they chop it up. They recognize it as foreign and they chop it up. And so together we've been working on, so if he designs constructs that will evade this essential immune response of the organism. But he's shown where he'll design a construct like that and perform, standard delivery and still gets no transformation. So it's like in that case, there must be a physical barrier because the organism, it should be able to uptake, the plasmid and use it. And so working together, we have cases where, we know we're opening pores. We also have assays that we can do to show that we're opening pores. So we have cases we know we're opening pores, so we know we're getting things in, but then you don't see transformation. And so we're working together and kind of pulling those things together so that you can come at it from at least those two angles, know that you're opening pores, and once it gets in, you want to have something that the cell can actually use, and it's not going to recognize as foreign. But to the second part of your question, thinking about tools, kind of moving forward, I'm definitely thinking about not just this system. I think maybe a criticism of myself and maybe others in our field, we tend to design these tools. You and I talked about this actually. We design these tools and then we keep using the tool over and over again. Yeah. And so I could care less how the delivery happens. I want the delivery to happen. So we're starting to look at, perhaps combining other physical ways, other things that have been published, because ultimately we just want to be able to get the information. And I think that there's actually an opportunity in coupling multiple things together, because classically there's been, there's an electroporation group, and there's groups doing mechanical things, and there's groups using ultrasound, and there's groups using chemical transformation. Many of these things are doing the same thing to the cell. They're putting some energy into the outer envelope. The envelope responds by opening pores. If you had more levers, I think you'd be better able to optimize for different cell types, particularly those that might be either very, very sensitive or just difficult to transform.
Yeah, my question's very related actually, along the lines of different mechanical stresses. Are these shear stress dependent, the viability results that you showed, and is that cell specific? Because I think there was a difference between the CAR T cells and the CAR NK cells in terms of viability. You looked at flow rate and how that affects the electroporation, but is there also an effect on viability?
There is for sure, and a couple reasons for that. Maybe the first order reason is just different cell sizes. Right, so the different cell sizes influence the electric field, not the electric field, but the effect of the electric field on the cell. So a smaller cell needs generally a higher field for opening pores. And so if you have two cell types and they experience the same field and one is larger, the one that's larger actually feels more stress. So to first order, there's that. But then also the cell type, I think that's an interesting question that we're looking to kind of dig deeper into, is understanding, is it something about the envelope mechanics? So more than just the size that might be affecting how they're responding to this.
My question is a little bit related to that, and I noticed in the sort of first section with the large volume transfection that increasing the flow rate seemed to decrease some of the transformation. I was just wondering if you had any more context for that, why that might be.
Yeah, that's a great point. And so in the first section, we're looking at bacteria, and bacteria are a little different because they also have a peptidoglycan layer, so they're a bit tougher. And we think a lot of the influence there is the influence on the residence time. So beyond a certain speed, they're just not perhaps spending enough time within the electrodes. Still enough time to open pores, but it's not optimal. So if we were able to perhaps make the channel longer, but then we'd need a different power supply, we're kind of stressing the limits of our power supply right now. So that's not a physics barrier, it's more of a practical engineering barrier in trying to show that we could perhaps use higher speeds, but then get the same field. And I think that's just a residence time thing. Because once again, if the flow rates were on the order of 2 to 3 meters per second at the very highest flow rates.
Last question.
Thank you for the talk. So I'm minimal electrical background, but I was curious. I know when you're doing DNA cloning and you're sampling your cloned products, your cargo is in a very salty solution sometimes. And I know that the increased ionic strength, when you don't dilute your cargo enough, it can really ramp down your characteristic time constant and you zap your cells dead. I was curious with the electric field requirements being decreased with the M2 platform, does that make your platform more robust against those sort of outcomes for cloning products?
Yeah, so that is an excellent point. And yes, so for example, transposons are often in very high salinity solutions. And the fact that we can reduce the field helps us there. So yes, so yeah, you're right on the money. Now, it doesn't get us all the way where we'd like to be. It's still tough to do some of those suspensions, but it's possible. And in some cases, we're able to do some things that you can't do in a cuvette because the conductivity is too high.
Great. Let's thank